for joining today. Oh, okay. Just heard it say recording in progress. So thank you everyone for joining today as I give this short tutorial on the use of GIGWA. My name's Daniel Edgegarza. I'm a postdoc over at the University of Queensland in Australia. It is now 6 o'clock in the morning for me, so I will get through this just having a little bit of a nap right before. So GIGWA is a genotype investigator for genome-wide analyses. And basically, if I can get this to operate, it's going to, no, okay, great. So it's a graphical interface that allows users to store and share large genomic data sets, filter the data sets, and export the subsets of that data in various formats for external applications. So those are the three main functions of GIGWA that I want to go through with you all today. So the data sets that I'm going to show you for the Rosaceae group is currently set up as one data set per project. And that was because when I began curating and putting these data sets up, GIGWA had only supported combining data sets with unique individuals, and it was always giving error after error if I tried to upload additional data to the same individual after I'd already uploaded it. So since that time, the developer has added a feature that allows us to upload information on the sample and connect each of those samples to the individual, which allows us a lot more flexibility with being able to upload additional data. So I will revisit going to these data sets and adding them to further refining them for the database. But as of right now, you can still access them publicly on GIGWA as a single data set per database and connected with information to its publications. There are several formats for the data sets to be uploaded into GIGWA. I'll show this in a minute. But you got like HapMap, Plink, Dartseek, Flapjack, VCF files. You can also interface directly with breeding APIs. All you have to do is just provide the URL to the dataset in one of those formats that's supported by GIGWA. And then it should pull that information directly over. However, the caveat there is just that you are not going to provide any more additional curating of that data set, changing any information in it. It's just pulling it directly over. If you need to do that, which is what I've basically needed to do with all of the apple and cherry and peach and things, these data sets, then I download these data sets, I start manipulating them, I've turned them into VCF files, and I've uploaded them to GIGWA that way. VCF files provide a little bit of flexibility in terms of multi-allelic data and including null alleles in the data set. So it allows us to be able to share those data sets in as original the format as they were presented to me compared to the other types of formats that are on GIGWA. So here is the home page of GIGWA before anyone logs in. Publicly accessible, these data sets that you see here in this list. So you can click on database, you can access any of these data sets, you can have a look at them. You see some of them that are duplicated here, say NT or they say AB. Basically, it means that the data set's been uploaded in two different versions, a nucleotide format where it's just the ACGT format and the AB format, which GIGWA doesn't allow a B allele. But Illumina, presents data as AB format sometimes. So we've just basically coded the B allele as G, and those will be labeled up when you access the data set. When you see it in the information page, it'll show that it's a AG format, which is basically to be translated to AB. If one wanted to upload their own data, they can do that. Even without an account, they could just simply select unmanaged data and click on import data to upload the data sets. The caveat to that is that if you don't have an account and you're uploading data, then it'll be available to you for about 24 hours and then it'll disappear. And if you want to do any more analyses, you'll have to re-upload it again and start again. So here's the page that you would be brought to if you were to click on Manage Data. Again, not having logged in, you will see here, can you see my mouse? I'm wondering, just give me a thumbs up if you can. But basically there's in this upper right corner next to Ploidy, you would not see a field for being able to enter in a new database. That's the one feature that you would not be able to do if you don't have an account. But you can still enter in taxon information for the species. You can enter in a project name, a new run, project description for your data set, the technology that was used, and enter in your data in any of the formats, be able to run analyses, do whatever you want with it, and download it, the information afterwards. And then after 24 hours, again, it's gone. So taxon information I've been entering in for the data sets. Once you enter that code, it'll bring up the species name. You can search for the species that you're entering. Floydie, I don't enter in the information. It usually GIGWA is pretty good at determining that from the data set. But basically, when you have the full version, when you do sign in, you see this extra field appear. This is where I've been adding new databases. And what I'm trying to get to within the next couple of weeks when the data sets are fully integrated is to integrate the projects by the ploidy of the data set. So if there's a 2N data set, they get all integrated together, and then there's 3N data sets. They'll get put together. So I'll have those as separate projects, and then the run will be each of the different sources. And so then all that information then will be described in the project description. Usually I would uncheck this skip monomorphic variants because it's easier to filter them out afterwards than to have to add them in. And then that extra feature that I said before that was added is that we can add the information based on sample now instead of the individual, and then link that later to the individual so that you can add additional metadata and multiple data points for each individual. And so that when you click this checkbox, you'll see that there's an additional file format down here at the bottom, an accepted extension TSV or CSV file where you can add your sample individual map. So basically, it's just two columns in the file, in this TSV or CSV file with the headers sample and individual. And it's just showing a list of all your samples to map it to whichever individual. Once you've uploaded whichever format you wish, again, for me, it's been VCF, so I click on VCF, I select the file, and I click on my map for the sample and individual and select that file. They'll appear here on the side to the right. You click submit. A green bar will start to go across. And if it works, which here lately it's been a little bit troublesome, but we're getting there, it will, once it's gone to completion, it will indicate that your files have been accepted and uploaded to the system. And then you'll start to see a percentage going up from 0 to 100, showing that all your variants or your markers are being processed and uploaded to the database. Once that's completed, then your database is ready. This is a pretty quick process, usually able to process, well, like 320,000 markers we're able to process in about 5 minutes on a university system. So that was pretty quick. And so that's that process. Then when it comes to the metadata, you click up here to the metadata import, and that will take you to this page. And if you do this right after you've uploaded the data, then your database will already be showing here, and you want to import the information on the individuals. And you'll then select a file here that will have one field, one column that is labeled individual, and then any number of other additional columns you wish for whatever other metadata you wish to have connected to that individual that you want to use to be able to subset or filter your data. So you make that file as, again, a TSV or a CSV file. You put that here and you upload it, and then now you'll have information, metadata information on your individuals. And I'll show how that works in a minute, how to be able to filter on that. So again, coming back to the home page, here's the list of all the data sets that are currently up there. You've got some Apple data sets, pear data sets, Rubus, Strawberry, and I plan to have additional data sets brought over from the GDR up here within the next couple of weeks. So GIGWA, its main function is to allow you to be able to filter and share data. And so there are two types of data that you can filter, and there's two ways for each type that you can do the filtering. So the two data types are the markers and the samples or the individuals. And then we'll just go into each of those. So the filtering on the markers, you can do that using the internal GIGWA tools. Here, it's showing that you can do that on variant type, which for our data sets, it's only been SNP, so there's really not a filter there. But you can also filter in addition to the SNP, you can filter by chromosome or any number of chromosomes. And you can also filter using a range um, along the chromosome and whichever markers you would like to filter within that range. So those are the internal features that you can do some mass sorting on. Then you can do additional manual sorting or manual filtering using the variant IDs. And so here you would check the box filter by variant ID, and then that's going to gray out the internal tools. And you can start typing. When you click on here, you'll get a drop-down menu of all your markers that are in the data set. And basically, you can start typing the markers you'd like to choose, and it'll filter them. You can start choosing any number of these. So here you can see that I've selected 2 of them. You can also copy these to a clipboard. You can paste them to an additional clipboard, or you can paste this from another file. If you were to copy it from a file as 1 marker per row, and then just copy it into here and click apply, then you can filter it that way. You see that there's 2 items selected here. This orange arrow now is uh, pointing to the upload button. If you were to click on that, you can just upload the file itself. And here you can see I've got a text file of common markers between the rose breed and the fruit podomics data sets. And so I've got them listed as 1 marker per row. I do not have a title or a header for that row. It's just markers alone. And when I load that in there, it updates to show that I've got now 2,131 variants selected. If I was to click on that, I'd get the drop-down menu again of all the variants that I have selected. I can do additional filtering by deselecting some of these if I want to refine that. And that's basically that for filtering on markers. So going back here, you can also filter the data in GIGWA based on the individuals or the samples using, again, internal tools or manually. So we'll start with the internal tools. To access the filtering on the individuals, you just need to click on this field here called disabled by the investigate genotypes. If you were to click on that, you get a drop-down menu here. Individuals shows that all 488 have been selected in this particular data set. If you were to click on individuals, you'll get a drop-down menu, which I think is not shown correctly on there, which there we go. There we go. Drop-down menu of individuals. You can then start to select on those and filter them. What I did a second ago, I know what I did right now, is I'm going to go back and I'm going to say that I was talking about internal again. This is basically selecting on the internal tools, the missing percent, MAF, minor allele frequency, and heterozygosity, sorry. So what that's doing here is if you wanted to like filter out individuals that have more than 20% missing data or 10% missing data, you can do that. You know, allele frequencies greater than 5%, to 50% if you wanted to keep those, but anything lower than 5%, filter that out, or heterozygosity of any range, you can do that. I've selected here, just filter out anything that's got less than 5% MAF, and it gives me, when I click search, it gives me then those markers that are then filtered on that. Those are the internal tools. So then going back to the manual ones, that's when I was saying about selecting on the individuals. So you get the drop-down menu, you can start typing to find out which individual you want to select, or a range of individuals. You can click on that, drag your mouse down, select them. You could do a couple of discrimination between the individuals that you want that you've selected. So here I'm selecting Macintosh. I just started typing in Macintosh. I selected that. I started typing in this WASIC Macintosh. Selected that. It shows that I've got two individuals of those 488 selected. And I've chosen a genotype pattern all different. So what this is going to do, since these are in the same group, is it's going to give me a list of all the markers that are giving me a different genotype for these two individuals. And so when I click search, then well, that's all different. Click search. And what I get are three markers out of the 3,000 or so in this data set that are different. And that makes sense because, I mean, WASIC is a sport mutation of the Macintosh. So you would expect that there'd be few differences here, and don't know if they're real, but these are the markers that you can take a look on. If I wanted to click on any of these, I can click on, for instance, the top one, and it'll give me the genotype for each of them to show how it's different. So it shows that the WASIC one is homozygous for the C allele, the Macintosh is heterozygous for TC at that particular marker. So uh, this page shows that you can also copy your selection. Yeah, so you can copy it using this tool here, the clipboard, and then you can also paste it to the clipboard, much like you were doing with the variants up at the chromosome level or the marker level, and you can save them. Now, that's all good for broad filtering, but another way that you can filter is manually by the names and on the metadata. And so if you've added a metadata file for your individuals, then you'll get an extra feature here, this magnifying glass. When you click on that, you then get this page that you can scroll along. And it's got all of your different metadata columns that you've added, along with your individual column that shows your individuals. And you can use then any of these metadata here to filter and click apply, and that will filter your data set for you in the different groups. So that's another way that you can use to filter your data. So I'm going to show one more differentiation analysis here by filtering along individuals. So in the fruit proteomics project, I've selected two groups. And in the first group, I've selected all these FUPI individuals. And in the second group, I've selected these GDR individuals. And then I'm going to take a look at both the internal and the manual tools here. So I'm going to say that I want to keep those individuals within each of those groups that's, got no more than 20% missing data. For the MAF, I'll just leave it as it is because some of them could be like, the group could be completely homozygous for an allele. The heterozygous percent, I'll just leave as heterozygosity, I'll leave as same as well. And the genotype pattern, I will select as all or mostly the same within the group, and then between the two groups, discriminate with the other group. So that then should give me a set of a set of markers that are of one type for one group, but are of a different type for the other group. And so when I click on search for that, I wait for it to finish, and it gives me then the list of markers where that meet that criteria. And this is giving me like 7,000 markers out of a 200 and 270,000 markers or so. So then I can click on any of these and view them as I did before, or I can, uh, here you can see that this is one of the individuals. So yeah, when I click on it, you can see that they're all homozygous for this A allele, and the other ones are heterozygous AC. In addition to viewing it that way, I can also click on these visualization charts at the top right of the screen, shown by this arrow here. And so when I click on that, I can start to play around with these displays. So I can look at the density of the markers that have been selected along the chromosome. I can take a look at FST. I can take a look at MAF. So here's the density along chromosome 15 of the markers. Um, yep. And then for the MAF distribution, if I can select that on both the groups or on one group, here's the distribution on both groups. Here's one group, and the other group is just the flip of this, so I won't show that. But basically, you can start to further refine your marker selection based on that. FST, well, because I selected the markers to be all the same within the groups, then I'm not going to get much variation in this, but you can still see that there's some deviation between the two groups there. And if I was to take off that filter of it being all the same and select all the markers that are within those groups, then I can start to look at some local differences along the chromosome. Um, so that's what you can do with this sort of display. And with the TAGMSD, it's a similar sort of situation. You can use that to try and determine if any selection, what's due to selection, what's due to random evolution. So there's probably not much going on here in terms of like targeted selection, but all the others, it seems like there's been a lot of selection going on. Take that out of the way. This hamburger menu up at the top, kind of hidden it right there. If you click on that, you can print any of these charts, and you can download them as images, documents, print them to a separate page, download the data actually for each of those points into a CSV or Excel file. So that's some of the features that you can do there. In addition to the charts, you can click on this integrated genome viewer, and you can load the reference genome when that page comes up. It's got a lot of, GIGWA has some preloaded genomes for you, but doesn't seem that Rosacea is on there. So you can load that from a file. I've downloaded the file either from the GDR or from the NRA website. You can access the file there. And then what you need to do though is use maybe BCF tools to make an index file from that, and then you can upload those together. It takes a second to upload really, and then you get the genome along there. And then you can load the track, which will give you the annotations of the SNPs along those chromosomes. So if you pull that information again from the GDR, from INRA or any other website that has this information, you can index that file again using BCF tools, upload these two files together, and then under the display genotypes, select either all your individuals, a group of them, or just any one of the groups, and start to zoom in. And when you zoom in, you can see then all your markers the genotypes for the markers that you had selected, for the groups that you selected, uh, along the chromosome. Since in this data set I had labeled each of the markers as just their chromosome and position, this then helps you to link it back to the marker name that's annotated to the file. The individuals are located over here. You can scroll alongside each of these to see all the genotypes that are color-coded. You can click on the level with each of the groups, any one of these markers to, um, well, this is the groups, sorry, had animation. These are the markers that you want to select if you want to obtain different information or additional information. So this, for instance, group one, this marker will give you the reference G allele, the alternate A allele. If you click on more info, it will take you back to the genotype page where it shows the genotypes between the two groups in detail. And then also the variant stats on the left side that show um, the stats for each of those markers. So it's just another interactive view that you can use to help interrogate your data. So finally, you can push a subset of the data out to different tools here or here. These have sort of been deprecated. It says as much inside the documentation. But basically what these were is, and they still work, though not very well, is if you provide a URL to JBrowse from the GDR or from INRA, then it will, um, when you click on the marker, it'll take you to that page, to the JBrowse at those sites. The only thing is though, is that when, in my experience, when I've clicked on them, it doesn't take you actually to this marker. If I click on this particular one here, for instance, it'll just take you to the page. And so it's not very helpful. I just usually stick to the integrated genome viewer, which the documentation is saying is superseding these things. So you kind of can potentially ignore these. But just in case you wanted to know what they were, now you know. This here, the configure online output tool, allows you to be able to push your data out to your favorite Galaxy instance, for example, or to other programs like the diversity analysis with SNPplay, which I haven't been able to go in depth to be able to take a look at. I've been trying to upload some data to it. It hasn't worked exactly. It doesn't seem to like the file for some reason, but when I get more info, I'll email it. But yeah, it's these SNPplays allows, like, supposedly has some different ways that you can view the data and subset the data. So it's just another tool. Once you're done with visualizing your data, filtering it, getting it the way you want, you can download that data into a new file. GIGWA supports many different export formats. I've been using VCF a lot to integrate data together into a new database. You can export all groups, which I selected group one, group two. You can have up to 10 groups. You can export all individuals, like the total. Even if you were viewing just those groups, you can select everything. Or then also you can export the metadata information if you wanted with those in the file, and it'll export it all as a zipped file for you. You can export it as a URL, or you can keep the files on a server, or both. Do know that if you keep the file on the server, it only keeps it for a limited time. I think I have a slide on that in a second. Basically, this is right here, this download tool. So you click on that. I'll show you, for example, Flapjack. So click on Flapjack. Basically, I'm selecting all the groups, group one and two. I've selected all my metadata. I'm going to keep it on the URL and on the file server. I'm going to click export, and then it shows me the Flapjack file. And the cool thing about this is that it's interactive. So as I'm scrolling along with my mouse, which I can't do here in PowerPoint, but as I'm scrolling along with my mouse, over each of the different markers, it will highlight that marker in red up at the top along the genome so you can see where you are. You can left click and drag, and you can move along the chromosome to see your data. Each of these are kind of like HapMap, where one allele is suggesting that it's homozygous for that allele, and then the heterozygotes are split like this. And then down here is just the broad view of, what's these broad patterns of homozygosity and heterozygosity that you can see among the alleles at the different markers. Uh, so you can use zoom to zoom in, you can zoom out. Yeah, all sorts of, and look on each chromosome right there. So this is the unincorporated contig there that we're looking at. So you can also export your data as neighbor joining tree. This Newick suggests that the tree is a nested um, series of distances inside parentheses that you then put into an online viewer or to another local program to view the tree. I'll chosen to use the integrated tree of life, interactive tree of life, ITOL, and this is all the data. It's not very visible, but when I turn it into a rectangular view, I can start to prune out information. Of course, it would have been better just to subset my data and then just import it directly as a subset, but um, yeah, there you go. This is the subset here, basically. And then I can turn that again into a circular view and see that how the tree is viewed related to each other, these individuals zoomed in. So and that was your tree view, and then BCF. So again, when I select a um, file format to export to, if I want to keep it on the server, you'll notice here it says the export file will be available at this URL for 1 hour. So there you go. As promised, I came back to that. You have 1 hour to download it, or you need to go back and do it again. And so that's that part for the downloading of data. Up here at the top, you see that there's a docs link. If you click on that, it'll take you to this page. If you click on the southgreen.fr, that's the developers, it'll take you to their page. It'll have some additional documentation there. It's an old page. Basically, what you can get from it is what you can read on the page when you get there. Any of the links are basically non-existent anymore. You can't access them. And I've contacted the developer about that, and he said that basically when they had migrated to a new server that they lost all of that, and it's on his wish list to get that all back up and running. And I told him I'd love to help out with that as soon as I have more comfort in um, my learning curve on learning all of this about GIGWA and some of the additional features and be able to access the full power. But hope I was able to show you guys at least the basics of being able to upload, filter, and download your data sets to be able to share. And with that, if you have any questions, I'm happy to try to answer them as best as possible. So starting to get light over here. New day.